Start.
Welcome. Myself, Dr. Prosanta Saha, working as an assistant professor at the Department of Botany, Durgapur Government College. And today I am here to deliver a short presentation on the restriction mapping of DNA. So, in this deliberation, I would be covering these topics. That is, firstly, there would be an overview of the process of restriction mapping, followed by some discussion on the restriction endonucleases, what they are, structurally and functionally, and thereafter I would be getting to the process of restriction mapping, followed by a very short web tutorial on how a restriction map is being prepared, and thereafter I will discuss the applications. So let's get started. So if we consider the word restriction mapping, it is composed of two words. First is restriction and thereafter is mapping. Restriction, which actually means to confine something. Restriction, that is confinement of something. Say we have a very big thing, we want to confine it into a certain things of shorter size or like that. And mapping, on the other hand, it relates to the positioning of something. So cumulatively understanding it, that if we consider the word restriction mapping, then that means we are there to position something or to understand the position of something based on its restriction or fractionation or limiting or its confinement to something. If we have a look at this diagram, we will see that there are landmarks given alongside, that is a way, pathway or a highway. Say for instance, we are moving from Kolkata to Bardhaman, we will first encounter Dankuni, thereafter Palsit, then Shaktigarh, and eventually Bardhaman. Now, these positions like Dankuni, Palsit, they are nothing but landmarks which actually emphasizes which position we are there at that moment. Similarly, if we look at this diagram over here, it shows a DNA sequence of some 100 kb length, which is traversed by certain colored lines like that. Now, what are these lines? These lines are nothing but the recognition sites of some restriction endonucleases. Restriction endonucleases I would be detailing in the subsequent slides, but they are certain enzymes which cleave the DNA. Now, if we consider the restriction endonuclease recognition site, say these are the restriction endonucleases like BAMH1, ZO1, NH1, BGL2, and they are being color coded differently. So, the restriction sites of BAMH1 would appear over here, then it is appearing over here and appearing over here and like that. Similarly, that of ZO1 we can see it is appearing over here and then here like this. So, the DNA positioning, the positioning in the DNA of the restriction sites of the different enzymes actually works to locate different positions of the DNA. So this is the essence of our topic today, that is restriction mapping of DNA. So as I mentioned in the previous slide, that is it is done by the activity of certain restriction endonucleases. So first we are here to understand what are restriction endonucleases. Now restriction endonucleases, again if we focus on the term, it consists of two words. First is restriction, that I have already mentioned, that is confinement of something, and thereafter it is followed by endonuclease. Again, endonuclease, we can cleave it into two words. That is endo, that means something inner, and nuclease. Nuclease means an enzyme which cleaves the nucleic acid. Now the endonucleases or the nucleases, they are basically of two types. One is the exonuclease, which can cleave a nucleic acid molecule, be it DNA or RNA, from one endo. Alternatively, there might be the endonucleases, which cleaves the DNA from within the DNA molecule. So these are the restriction endonucleases. They are certain enzymes which have the recognition site within the DNA and they cleave the DNA not from its ends but from the between one. Considering the types of restriction endonucleases, they can be categorized into four basic types, that is type 1, type 2, type 3 and type 4. Out of which the type 1 and the type 3 enzymes, they recognize a point of DNA but they cleave DNA at some other point. On the other hand, the type 4 enzymes, they are specific to certain modifications of DNA like methylation. But if we come to restriction type 2 restriction endonuclease, we will find that these enzymes, they cleave the DNA at the point of the recognition. That means they will recognize a certain sequence of the DNA and cleave it there. Now, the recognition sequence of this type of enzymes, they are basically palindromic in nature. Now, what are palindromes? The palindromes are such sequences which will spell and read the same irrespective of the fact whether we are reading it from the start to the end or from the end to the beginning. For instance, if we consider the letters of the word Malayalam, it is spelling and reading the same irrespective of the fact whether we are reading it from the start to the end or from the end to the beginning. Similarly, the restriction endonuclease sites, they are palindromic in nature. For instance, if we have a look over here, the first strand, in this strand, the first sequence that has been shown over here, that reads CCCGGG in the 5' to 3' strand. And in the antiparallel strand of the DNA, that means this would be the 5' end and that would be the 3' end, it again reads the same thing, that is CCCGGG. Overall, the sequence reads like this: CCCGGG, CCCGGG. Now, these are the types of sequences that are recognized by the restriction endonucleases, specifically of the type 2 restriction endonucleases. Now, following the recognition, as I mentioned, the type 2 restriction endonucleases, they cut within the sequence that they recognize. So, the nature of the cut produced by the, or nature of the digestion that is produced by the restriction endonucleases, that can be of two types. Either it might be of the blunt end type or it might be the staggered end type. Blunt end type means, see, there is a clear cut, no overhanging DNA sequences. Whereas on the other hand, it is producing the staggered cuts, there we are having certain overhanging sequences. So here there is presented certain examples where which do either blunt end cutting or the staggered end cutting. There are different types of restriction endonucleases are shown. And then again, one other thing that I would like to mention over here, that the nucleotide recognition sequences, that might be either 4 nucleotide sequences or it might be hexanucleotide sequences. It might be octanucleotide sequences and like that. But the tetranucleotide sequences and the hexanucleotide sequences, they are relatively more common. So here's an example provided over here with respect to the enzyme Iqac-R1 and its recognition site. One thing I should have mentioned that these restriction enzymes of the restriction endonucleases, they come from the microbial sources, basically from the bacterial sources. So like here, the Iqac-R1, if I emphasize on the name, this being an enzyme, so it comes from the bacteria Escherichia coli. So E stands for Escherichia, CO stands for coli, strain R, and the first restriction endonuclease from that source. So it is Iqac-R1. So like here, the naming is done in such way. And this, the Iqac-R1 enzyme, that actually recognizes this hexanucleotide sequence, that is GAATTC. Again, as we can see over here, it is a palindromic sequence, that is GAATTC from the 5' to 3' of the sense strand, and in the antisense strand also it is reading GAATTC again. And following its recognition of this sequence, this enzyme, it makes a staggered cut over here. So this enzyme, it binds over here and it cleaves the DNA following a cut that is producing overhanging sequences. That means what? It is producing a staggered end cut. So coming to the technique of how now, till now we have studied that how does the restriction endonucleases work, very briefly. Now what are the techniques that are involved in the restriction mapping? So there are basically two techniques that are involved in the restriction mapping of DNA. First, we need to cleave the DNA by restriction endonucleases, and thereafter study the cleaved products by agarose gel electrophoresis. A flowchart has been shown over here which shows that first a sample DNA is taken, then thereafter restriction dilations are performed. Now, at least 2 2 enzymes should be involved. If not more, can also be taken into consideration. But at least 2 enzymes should be involved, and in this case, both the enzymes will first be used to cut the DNA individually. Like for instance, if I take enzyme A and enzyme B, first I will cleave the DNA with enzyme A only. Thereafter, I will cleave the DNA with enzyme B only. Following that, a combinatorial digest is done. And in the combinatorial digest, a combination of both enzyme A and enzyme B are together used to cleave the DNA. And the digestion products, they are thereafter run on the agarose gel, and following by comparing the bending profile of the produced by the individual digestion by the enzymes to that of the combinatorial digest, we will elucidate the position of the restriction sites into the DNA molecule. So this is a very brief overview of the technique that is being done. An example is shown over here. Over here, like that, we have a 12 kb DNA fragment that is to be mapped. So what is to be done? First, I will cleave it with the first enzyme that is taken over here, that is EcoR1, and it produces 2 fragments, that is of 3 kb and 9 kb. That means the 12 kb DNA fragment has been cut into 3 kb and 9 kb fragments. Second, I will take another molecule of the same DNA and I will cut with that of the another enzyme, that is my enzyme 2, in this case BAMH1. BAMH1 cleaves the DNA molecule into 2 fragments of 5 kb and 7 kb sizes. See, first enzyme EcoR1, it cut the DNA into 3 kb and 9 kb. The second enzyme, that is BAMH1, it cleaved the DNA into 5 kb and 7 kb. Thereafter, in the third step, what I said, that we will do a combinatorial digest. Over here, that is mentioned over here, that is we are doing a double digest or a combinatorial digest with employing both the enzymes simultaneously. And when we do it, what we see? The bending profile changes. Here the bending profile shows 2 kb, 3 kb and 7 kb. Now, when we run these digestion products into agarose gels, somewhat like this, the bending profile, it is being shown. On comparing the bending profiles, we see that the 7 kb of DNA that was produced, 7 kb of the fragment that was produced following BAMH1 digestion, it is persisting in the combinatorial digestion also. The 7 kb fragment is found over here as well. However, the 5 kb fragment that was produced by the BAMH1 digestion is no longer available in the combinatorial digest. Instead, we are finding that 2 fragments are being formed, one of 2 kb, another of 3 kb, the cumulative size of which amounts to 5 kb. Again, on comparing this combinatorial digest with the EcoR1 digestion profile, we find that the 3 kb fragment is persisting over here, like that of the EcoR1. Now, what does it show? It shows that the 5 kb fragment of the BAMH1 produced by the BAMH1 digestion is somewhat intervened by an EcoR1 site. So when the combinatorial digestion is done, the 3 kb site is coming from the first end, the 3 kb site is moving from the first end, and thereafter a 2 kb of remaining 2 kb of the BAMH1 digestion, followed by the last fragment produced by the BAMH1, that is 7 kb of size, that is persisting in the combinatorial digest as well. So if we now locate the size of the size of the sequences into the DNA molecule, we will see it like this: a map of 12 kb DNA having EcoR1 at the 3 kb and the 9 kb sites, and BAMH1 at the 5 kb and the 7 kb sites, right? And on the combinatorial digest, we will be having a 3 kb, thereafter a 2 kb, and finally the 7 kb. So this is all. And all these are studied by running them into the agarose gel. And in the case of agarose gel, as we all know, that DNA being negatively charged, it migrates from the cathode to the anode. So the bigger sizes of the fragments, they would be on the top side, and the smaller the fragment is, it will migrate towards the bottom. So the lesser size fragment like the 2 kb, that will come towards the bottom, then On top of that, there would be the 3 kb fragment, and the 7 kb fragment would be just right on top of everything. Now, this process, it can be studied, or the restriction map can be generated using several computer web-based servers. So I will be just, as I mentioned, a short web tutorial would be provided. So I will be just getting into that. But just before entering, I will just give you an overview of what is there to be discussed. First, we will retrieve the sequence of any DNA molecule of our choice. That we will get into the NCBI website, that is the National Center for Biotechnology Information. From there, we will type the nucleotide and we will type the sequence of our choice. And when the sequence comes, we will open a FASTA file that will be shown. We will copy the file. Next, we will again open another tab, and there we will be opening another server, that is NapeCutter. We will paste the copied sequence over here, and then first we will do the first dilation with the first enzyme. We will note down the fragment sizes that are produced. Thereafter, we will digest it with the second enzyme and note down the fragment sizes again. Following that, we will do the combinatorial digest, and thereafter we will again note the fragment sizes. Then finally, we will compare the sizes of the fragment, and thereby we will create the restriction maps. And these are like the screenshots of the servers that we would be working with. That is first the NCBI that is shown, and in the bottom side that is the NapeCutter that has been shown. Just before getting there, I will just like to emphasize on one thing. As I mentioned that the DNA fragments, the smaller size fragment that migrates towards the bottom, and the bigger size fragment that being on the top. So the theme of the experiment is that digest with enzyme 1, digest with enzyme 2, make a combinatorial digest, compare the fragments. But over here, what is shown that enzyme 1 digestion producing 2 fragments. Enzyme 2 digestion, it has been shown that 3 fragments, 3 orientations are shown, out of which 2 have been cut and this has been selected. So this, how it is done? This is done by just comparing the band sizes with that of the combinatorial digest. I will get back to this slide, but before that, I will just have a very short web-based tutorial. So over here in the NCBI, that is the NCBI site, as I mentioned, first I will get into the nucleotide. Once inside so this is the interface, looks like that. Once into the nucleotide, here I will type a sequence of my choice. That is, say I am typing DREB1A, DREB1A. I will click search. See over here, several pages are opening, several links are opening. I will just get into the first link. That is the Arabidopsis thaliana gene for DREB1A. And over here, like I mentioned, that I would be taking the FASTA sequence. Why? Because see, the normal sequences, as it is shown, that is not continuous. There are certain gaps over here, like and the numbering of the nucleotides are also present over here. But if we take the FASTA sequence, it will be a continuous sequence. So this is how the FASTA sequence looks. And over here, I will copy the sequence. So I'll just copy the sequence, and then I'll get into, say, NapeCutter. So inside Google, I'll just type NapeCutter V3 and an interface like this will come up. So if I just paste the sequence over here and name the project, like say I just want to name it suppose P1. Okay, so just a minute. So I have named the project as P1, and I have copied the pasted the sequence that I just copied from the NCBI website pertaining to the grade 1A gene. Then I'll click submit over here. Now you see what happens, as is a known piece of DNA, so already there are several sequences of the maps are provided over here. But for the demonstration purpose, if I want to select at least 2 enzymes are required for the creating the restriction map. So if I, for instance, if I want to select just 2 enzymes and create or locate their positions into the DNA and thereby create a map of the restriction map of the DNA out of those 2 enzymes, what will I do? I will first go to custom digest. Custom digest means I am customizing the digestion pattern. So over here it is shown, several enzymes are shown over here. Say I will select the first enzyme like ACC1 and I will click on digest. Now as we can see over here, there are 2 sites of ACC1 digestions. One is at okay, so here are just 2 sites of ACC1 digestion. So 2 sites of ACC1 digestion means there would be 3 fragments, right? So if we look into the gel profile over here, we will be finding the gel will look like that. And the fragment sizes would be of, the first one would be from 1 to 1913, that is the length would be 1913 BP. The second one would be from 1914 to 3305, that is the length would be 1392 BP. And the final one would be from 3306 to 3334, that is the end of the DNA molecule. That is the very short fragment, that is just of 29 base pair length. So the next thing is that I have to take a note of these fragment sizes. So following ACC1 digestion, I am getting 3 fragment sizes of 1913, 1392 and 29 base pairs. Thereafter, what I will do, I will go again to custom digest. First time I digested with my first enzyme of my choice. Now I will be digesting it with the second enzyme of my choice. So I will deselect this one, right? I will deselect this one, select another enzyme of my choice, like over here I am selecting say BSAH1 and I will click digest. BSAH1 it cleaves only at a single point. That means how many fragments would be there? 2 fragments would be generated. So if again I look at the gel, so this would be the gel image line, right? 2 fragments are generated. The first fragment is from start to 2960 BP. So the length is 2960 BP. And the second fragment, that would be from 2961 to 3334. That is the length is 374 BP. See, a smaller fragment 374 and the larger fragment that is of 2960 BP. Now coming to the last step. Last step is what? Combinatorial digestion. I have to keep a record of these 2 fragment sizes as well. Now, in case of combinatorial digestion, my BSAH1 has already been selected. And what is the other enzyme that I worked with? That is ACC1. So I will select ACC1 also. So 2 enzymes have been selected now. And now I will do the custom digest with these 2 enzymes. And see, this is the profile like. What would the gel look like? When I look into the gel, what I find that the first point, that is the first fragment generated by ACC1 is intact, that is of 1913. However, the second fragment generated by ACC1, that was of 1392 bases, it has been cleaved into 2 fragments of 1047 BP and 345 BP. Which indicates what? That the BSAH1 site was lying somewhere between the 2 ACC1 fragment sites. And the last, again, the last ACC1 fragment that is also intact. that is of 29 dB. Now, if we look at the say graphical view, that is the map that is being generated, we are seeing that the two SEC1 sites have been intervened by the BSA H1 site. That means what? Here, by our custom digestion, we can generate a map of this DNA molecule by customizing our digestion with these two enzymes, and we can map them or locate them onto the DNA molecule. So this is how the restriction mapping that is done. So I'll just get back into my presentation. So that means what? That I just shown that we select two enzymes, do individual digestions, thereafter we do a combinatorial digestion, just compare the band sizes, whichever band size is in that means that fragment is not intervened by the another enzyme. Otherwise, whichever fragment has been intervened, that means there the other enzyme site is located over there. And then we have to arrange the orientation of the sites. Like over here, it has been shown that enzyme 2 restriction, it can produce the yellow, green and the red color portions of the DNA in this orientation or this orientation or this orientation. But it has to be the band profiles, matching the band profiles, we will come to understand that this is the correct orientation. So this is how restriction mapping is done. And what are the applications of restriction mapping? They find a wide range of application in molecular cloning, from identification of the positions of restriction sites to the study of insertion and deletion sites, from the mutational study, then verification of the size of the insert during cloning experiments, and analysis of insert during any transformant analysis as well. So these are the sources that I used for preparing this presentation. Thank you for your patient hearing.